As part of the Megazyme product range, Reagent provides an extensive range of assay kits for use in various assay formats, including autoanalyzer, microplate, and manual spectrophotometer. This lactose and D-galactose test kit, K-LACGAR, is a simple method which allows for the rapid measurement of lactose and D-galactose in food and plant products, and was adopted as an AOAC official method in 2006, AOAC 2006.06. This video tutorial will demonstrate the use of the kit with a milk sample, using the manual spectrophotometer format. The kit contains sufficient reagents for 115 assays. And a detailed assay protocol is available to download from the product page on the Megazyme website. The principle of the enzymatic reaction involved in the measurement of lactose is shown in this figure. Lactose is hydrolyzed to D-galactose and D-glucose by beta-galactosidase at pH 5.0. Interconversion of the alpha and beta anomeric forms of D-galactose is catalyzed by galactose mutterotase, GAL-M. This step is paramount in ensuring this normally rate-limiting step can be catalyzed rapidly, allowing for rapid analysis times. The beta-D-galactose then formed is oxidized by NAD+ to D-galactonic acid in the presence of beta-galactose dehydrogenase at pH 8.6. The amount of NADH formed in this reaction is stoichiometric with the amount of lactose. It is the NADH which is measured by the increase in absorbance at 340 nanometers. Prior to sample analysis, prepare the required additional reagents, as described in the assay protocol. Subsequently, once all of the additional reagents have been made, the kit components should be prepared, and once prepared, they are ready for use in the assay procedure. Bottles 2, 5, and 6 are used as supplied. Dissolve the contents of bottle 3 NAD+ in 12 ml of distilled water and mix thoroughly. This solution is stable for approximately 4 weeks at 4 degrees Celsius, or can be aliquoted into appropriately sized aliquots and stored below -10 degrees Celsius. The contents of bottle 1, buffer, are diluted to 24 ml with distilled water by adding 21.5 ml of distilled water to the bottle. Cap the bottle and mix this solution thoroughly. This solution is used to dilute the entire contents of bottle 4 and should be used immediately. Transfer the entire contents of bottle 4, 1.2 ml of beta-galactosidase enzyme, to the 24 ml of diluted buffer in bottle 1. Cap the bottle and mix thoroughly. This is now solution 4. Divide into appropriately sized aliquots and store below -10 degrees Celsius. In this demonstration, we are using a regular milk sample with the standard assay procedure, along with the supplied galactose standard and an associated assay blank. Prior to sample analysis, the milk sample must be clarified for use in the assay procedure. Examples of sample preparation methods can be found at the back of the assay protocol. Accurately pipette 1 ml of milk into a 100 ml volumetric flask containing approximately 60 ml of distilled water. Mix thoroughly and incubate at 50 degrees Celsius for 15 minutes, with occasional stirring. After 15 minutes at 50 degrees Celsius, add 2 ml of COAs 1 solution and mix thoroughly. Add 2 ml of COAs 2 solution and mix thoroughly. Add 4 ml of 100 millimolar sodium hydroxide and mix vigorously. Dilute volume with distilled water. and mix thoroughly. Filter an aliquot of the solution through Whatman No. 1 filter paper. Discard the first few millilitres of filtrate. Use the clear filtrate sample solution in the assay procedure. Typically for milk, cream and yogurt, no further dilution is required and a sample volume of 0.2 millilitres is satisfactory. To simplify the COAs sample preparation procedure, a COAs clarification assay kit, K-LACGAR, is now available to purchase from Neogen's Megazyme product range. Visit megazyme.com for more information. Follow the manual assay procedure as described in the assay protocol. In this demonstration, we are following Procedure A, the standard assay procedure. This assay requires each sample to be applied to two assays, a lactose LAC assay for measurement of lactose and a galactose GAL assay for the measurement of galactose. A blank reaction and a standard reaction must be performed with each batch of samples in both the lactose and galactose assays. In this demonstration, we are using one sample tube, one LAC and one GAL, one blank tube, BL-LAC and BL-GAL, and galactose standard tubes, ST-LAC and ST-GAL, for each assay. For the standard tubes, we are applying the galactose standard to both assays. This applied standard in bottle 6, D-galactose, is a pure standard. Therefore, it is not necessary to test this standard in the lactose assay, but for demonstration purposes we will. Testing the D-galactose standard serves as a tool to ensure that the kit is functioning correctly. A prepared lactose standard can be used to confirm the effectiveness of the beta-galactosidase hydrolysis reaction. This standard is not supplied with the kit, but can be applied to the assays in a similar way. Pipette 0.2 millilitres of clear filtrate sample solution directly to the bottom of the sample assay tubes, one LAC and one GAL. Pipette 0.2 millilitres of the supplied D-galactose standard, bottle 6, directly to the bottom of the ST-LAC and ST-GAL tubes. Pipette 0.2 millilitres of the prepared solution 4, beta-galactosidase, which has been prepared in bottle 1, directly to the bottom of the lactose assay tubes only. When all of the components have been added, mix the tube contents by swirling and incubate the tubes at approximately 25 degrees Celsius for 10 minutes. Add 2 millilitres of distilled water to all assay tubes. Pipette an additional 0.2 millilitres of distilled water into the lactose blank assay tube, BL-LAC, the galactose sample tube, 1 GAL, and also into the galactose standard tube in the galactose reaction, ST-GAL. Pipette an additional 0.4 millilitres of distilled water into the galactose blank tube, BL-GAL, thus ensuring that the final volume of all assay tubes is the same. Pipette 0.2 millilitres of buffer bottle 2 into all assay tubes. Pipette 0.1 millilitres of NAD+ bottle 3 into all assay tubes. When all of the components have been added, mix the tube contents thoroughly and incubate the tubes at 25 degrees Celsius for approximately 3 minutes. After 3 minutes, record the first absorbance reading, A1, at 340 nanometers for all of the assay tubes. In this demonstration, we are using the Megaquant wave spectrophotometer, set to read at 340 nanometers. Alternatively, a recording spectrophotometer with 1 cm path length cuvettes can be used. Swirl to mix the contents of bottle 5 beta-galactosidase dehydrogenase and galactose mutarotase prior to dispensing. After recording the A1 absorbance value, pipette 0.02 millilitres of the contents of bottle 5 into all assay tubes. Mix the tubes thoroughly and incubate the tubes at 25 degrees Celsius for 5 minutes to allow the reaction to go to completion. Record the absorbance reading A2 for all of the assay tubes. This absorbance reading completes the lactose and galactose assay procedure. The absorbance readings A1 and A2 of the sample and the blank reactions are used to calculate the lactose and/or galactose concentrations in the original samples. When performing this test on the megaquant wave spectrophotometer, the absorbance mode feature must be used. The results will be printed via the onboard printer, or the data can be exported to a computer using the SF Capture software. Please see our megaquant wave video for further details. In absorbance mode, the results output are raw absorbance values for both blank and samples. Therefore, the calculations of lactose and/or galactose content can be performed manually as described in the calculation section of the kit booklet. Alternatively, we have also developed specific Excel-based MegaCalc applications for each kit of the Megazyme range to allow quick and easy results analysis. Results can be analysed using the MegaCalc application specific to this lactose and galactose assay kit, which is available to download free of charge from the Megazyme website. The MegaCalc spreadsheet provides full instructions for use. Open the MegaCalc worksheet and input the following: sample details, absorbance readings for the blanks. For each sample, input the sample identifier and the absorbance values for the samples. In this demonstration, the samples and standards have been applied to both the lactose assay and the galactose assay, and therefore will have corresponding data for each assay. In the case of the galactose standard, as it is 100% pure, it would have also been sufficient to just test it in the galactose assay. Alter the sample volume if a volume other than the default 0.2 milliliters is used. If dilution of the sample has been performed, then input the dilution factor used. If no further dilution was performed, the dilution factor is 1. When all of the data has been entered, the concentration of D-galactose and/or lactose in the sample is automatically calculated and given as grams per litre in the original sample. For solid samples, input the concentration of the original sample extract in grams per litre. The concentration of D-galactose or lactose in the solid samples is then automatically calculated and provided as grams per 100 gram in the original sample.